Hey everybody, welcome back to Environmental Organic Chemistry with Dr. Lisa. Well, this is it. We've reached the end of the semester. Can you believe it? I'm doing these recordings in 2020 because of COVID-19. We're all stuck at home. And maybe in future years you'll listen to this and you'll laugh and say, oh yeah, remember that? No big deal. We got through that, no problem. But right now I'm about ready to scream because I've been stuck in my house for 7 weeks with nowhere to go. So, yeah, good times. Time passes. This recording will stick around for a long time. And you've made it through the semester with me, Dr. Lisa, doing environmental organic chemistry. So the purpose of this final lecture is to just give you a summary of all the different chemical reactions that we've talked about in the second half of the class. And this will help to prepare you for the final exam. Every year on my final exam, I give you a bunch of different chemical structures, and I ask you, what reactions will this chemical structure undergo? And it's up to you to be able to apply what you have learned to each of those structures and tell me how it's going to behave. So, first, it's not working. Trying to change slides. Not working. Let's try this again. There we go. Okay, it's just slow. So the first chemical reaction we talked about was acids and bases, acid-base reactions. So when you look at a chemical structure and you ask yourself, could this chemical act as an acid or a base, you look for a couple of structural features. In terms of acids, you look for phenol groups, carboxylic acids, and then some groups that contain sulfur. So this SO3H, that's a acid-base functional group, can lose that hydrogen. And some thiols, a thiol is an SH group. And not all of them are great acids, but especially if this thiol group is attached to a benzene ring or something, then this hydrogen becomes a good, can leave and give you the negatively charged sulfur, and the chemical will act as an acid. For bases, you're mostly looking for nitrogen in the form of an amine, right, which is the NH3 group, basically an sp3 hybridized nitrogen, or a nitrogen that's stuck in a ring, but not nitro groups. Nitro groups do not act as acids or bases. So this is the slide we had for the acid-base lecture, and it was just pointing out, here's your carboxylic acids over here, here's one, here's one, here's one. So anytime you see this carboxylic acid group, that can be an acid. Here's a bunch of examples of phenols. The OH is attached to the benzene ring. That's what makes it a phenol. So phenol, 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 phenol. And then some of these sulfur-containing groups. So this is naphthene sulfonic acid. This is the sulfonic acid part. And then this is an example of a thiol. Just thiol by itself has a relatively high pKa, so maybe not very environmentally relevant. But this is thiophenol, and thiophenol does have an environmentally relevant pKa of 6.5. Regular old alcohols, not so much. Their pKa is well above 14, so they really don't act as acids. And then to determine whether a chemical is a base, you look for the nitrogen. You ask yourself, does this nitrogen in this chemical have a lone pair of electrons? If so, that lone pair of electrons can pick up a proton, and so this can act as a base. So again, here's some examples of amine groups. In these examples, these are all attached to aromatic rings. Here's one which is not NH2, it's nitrogen with these two methyl groups and then attached to a benzene ring. Here's trimethylamine. Here's an end of the terminal amine, end of the ring, end of the chain amine. And then piperidine is nitrogen inside a ring. And then here's all these other heterocyclic nitrogen here. So nitrogen inside a benzene ring, nitrogen inside a naphthalene, nitrogen inside a 5-membered ring. So anytime you see nitrogen inside a ring or as part of an amino group, then it can act as a base. Okay. Then we get to other types of chemical reactions. Basically, we can divide them all into two types. One is where there's no change in the oxidation state, and so that's usually hydrolysis and nucleophilic reactions. And then the other types involve some sort of change in the oxidation state, And when you have a redox reaction, you can have reduction or you can have oxidation. So there's both types. Reductive reactions in the environment are going to occur in anaerobic environments like sewers and landfills and salt marshes and deeply buried sediments, places like that. And so one of the many important forms of reduction is reductive dehalogenation. And then you can also have reduction of nitro or sulfoxy groups. And all of these redox reactions can be either biological or abiotic. Then oxidation, we talked about more in terms of photolysis. We talked a little bit about abiotic oxidation of a few specific chemicals, mercaptans, anilins, and phenols. But really, this is the important part. We talked a lot about photolysis. And there is some aerobic oxidation, which is biodegradation. We didn't really talk about that this year, but in other years we have more time, and so we can have a lecture on biodegradation. So in terms of reactions that are not redox reactions, the one that we talked about was hydrolysis. The hydro here implies that the nucleophile is either water or hydroxide. So hydrolysis reactions are a subset of nucleophilic reactions. And we talked about SN2 reactions. These are the kinds of things that you're very familiar with from your organic chemistry class, and it's all review. Right, so for example, classic example, methyl bromide reacts with water to give you methanol. You have a proton left over here, and the bromide leaves as a Br minus bromide. So no charge on this side of the reaction. +1 and a -1 over here means no net charge on this side of the reaction. So there's no need to add any electrons to this reaction to make it balance. And therefore, this is not a redox reaction, which is how you know it's a hydrolysis reaction. So this SN2, SN1, and SN2 type of reactions occur on aliphatic carbons, right? Aliphatic carbons are carbons that have 4 bonds to 4 different other things, and they are what we call sp3 hybridized carbons. Now, to have an SN1 or SN2 type reaction, you need a good leaving group. We talked about which ones are the best leaving groups. Typically, you want these to be on a primary or a secondary carbon if it's an SN2 reaction. And you need some sort of nucleophile. Water or hydroxide is a reasonably good nucleophile. And there are other nucleophiles that might be important, like reduced sulfur species and things like that. But mostly for our purposes, we're thinking about water as the nucleophile. The other hydrolysis reaction that we talked about a lot was dehydrohalogenation, right? So dehydro means, D means you're losing, hydro means you're losing a hydrogen, and halogen means you're losing a halogen. So you're losing both a hydrogen and a halogen. And the hydrogen is leaving as a proton, HX plus, and the halogen is leaving as a halide, X minus, and so there's no net charge. You add those two together. So here's an example, 1122 tetrachloroethane. Hydroxide comes in as the nucleophile, attacks, kicks out one of these chlorides, which ends up over here as a chloride. But it's attacking at the proton here, attacking right there at that little proton, pulling it off. The electrons that were holding that proton in place swing in here to create the double bond. And then one of these chlorides has to leave. And you get, in this case, trichloroethene, chloride, and a water molecule. Since you started with hydroxide as your nucleophile here, you have a -1 charge on the left. You've got a -1 charge on the right, so there's no net change in charge. There's no need to add any electrons on either side of this reaction to get it to go forward. So for this reaction to happen, you need to have that hydrogen and that halogen on adjacent carbons, aliphatic carbons. Again, the carbons still, on this side of the reaction, the carbons have to be sp2 hybridized, right? On the other side of the reaction, they end up, here, let's erase this. On the other side of the reaction, they end up as a double bond, but over here, they're sp3 hybridized. So to get any of these hydrolysis reactions to go, you need an sp3 hybridized carbon. Then there's another type of hydrolysis we talked about, which was hydrolysis of carboxylic and carbonic acid derivatives. And that was this whole host of different structures here, but they all start with something with a double bond to something else up here. And, again, I don't expect you to memorize all of these different functional groups, but the phosphoric acid esters are a dead giveaway. These are very frequently found in organophosphorus pesticides, all of these things here in this bottom row. And then you got all these, carbamate. There's a whole class of pesticides called carbamate pesticides. And then these anhydrides are components of phthalates, which are used as plasticizers to make plastic flexible and pliable. So these types of functional groups are very commonly encountered in environmentally relevant chemicals. So you want to look for that structure, and when you see it, you think, that structure could undergo hydrolysis. The hydrolysis mechanism here is where the nucleophile, let's say it's hydroxide, comes in and attacks at that carbon. There we go, HO minus comes in and attacks right there. And then this thing is the leaving group. So just try to look for those structural features. Okay, in terms of rates and products, remember that all of these hydrolysis reactions tend to have definitely a neutral mechanism, and by neutral mechanism that means that water is the nucleophile. They frequently also have base catalyzed mechanisms where hydroxide is the nucleophile, and for these carbonic and carboxylic acid derivatives, they also have an acid catalyzed mechanism. So you have to worry about the acid, the base, and the neutral mechanism. Think about whether one or all three of those mechanisms are operating. And then in terms of predicting the leaving group, the leaving group with the lowest pKa will be the most favorable one to leave. So you can go back to your acids and bases lecture and look at what those kinds of pKa's are for those leaving groups, and that will tell you which one is the best leaving group. Okay, so that's finished our conversation about hydrolysis reactions, which are not redox reactions. Now we're going to talk about reduction reactions. The first one we learned was called hydrogenolysis. So it's very easy to confuse this with dehydrohalogenation, but it's different. This is hydrogenolysis, and hydrogenolysis is where you add 2 electrons to the molecule and a proton, and you end up replacing one of these chlorines with a hydrogen. So you take, in this case, carbon tetrachloride is being converted to chloroform. You get a chloride, so over here you have a +1 for the proton, over here you have a -1 for the chloride. So to get this thing to balance, you need to add your 2 electrons here. And that's how you know it's a redox reaction. Basically, anytime you see a halogen, whether it's aliphatic or vinyl or aromatic, doesn't matter, these can all undergo hydrogenolysis. So one of the classic examples is tetrachloroethene, PCE. PCE can be reduced to this, in this case it's being shown reduced to trichloroethylene, or TCE. TCE then gets reduced down to dichloroethylenes. There's 3 different isomers, cis, trans, and 1,1-dichloroethylene. And then the dichloroethylene can get reduced down to vinyl chloride, but the vinyl chloride is the most toxic of that whole chain of products, and sometimes the reaction stalls out at vinyl chloride, and this keeps countless environmental scientists occupied and earning good livings and putting their kids through college because of that fact that this often stops at vinyl chloride and won't go to completion. And so this is an aliphatic chlorinated compound. This is a vinyl chlorinated compound, a vinyl chloride, and this is an aromatic chloride, and it also can get reduced. In this case, you're looking at chlorobenzene being reduced down to benzene. In all of these cases, you got 2 electrons, 2 electrons, 2 electrons, 2 electrons changing hands, and you're adding a proton, 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 and you're leaving, the leaving group is leaving as an anion, bromide, chloride, chloride. So to determine, it's pretty easy to determine whether compounds are capable of undergoing hydrogenolysis. You just look to see, do they have a chlorine or a bromine? And if the answer is yes, then they can undergo hydrogenolysis. Period. End of story. Reductive elimination is more thermodynamically favorable. If a compound can undergo reductive elimination, it will, preferentially over hydrogenolysis. But that only happens where you have two halogens that are on adjacent carbons. When you have that kind of structure, as you have here, this is hexachloroethane. So you have two chlorines, one here and one there, on adjacent carbons. We actually have some extra chlorines too, but we have at least one on each of these two adjacent carbons. If you add two electrons here, you can kick two of these chlorides out and create a double bond. And notice we have two negatively charged anions over here, so to get this equation to balance, we have to add the two electrons here, which is one of the ways that we know that this is a redox reaction. So if a compound can undergo both hydrogenalysis and reductive elimination, elimination is almost always preferred. Then there are a few functional groups that can be reduced that are not halogenated. One of them is the nitro groups. Very, very common to find nitrobenzenes in the groundwater that are getting reduced to aniline. Again, you're taking these two oxygens off, and those two oxygens are ending up over here as water molecules. So to make them water molecules, if you have two oxygens that you want to turn to water, you're going to need 4 protons. Plus you've got the 2 protons that you're adding right here onto the nitrogen, so that's a total of 6 protons. And so we got a +6 charge over here and no charge over here, so to make everything balance we have to add 6 electrons to make this equation work, and that's how we know that this is a redox reaction. So nitro groups, very, very commonly reduced. Some of these sulfoxy groups also can get reduced down to more reduced sulfur species. So to determine whether a chemical can undergo this type of reduction, you just look for the nitro groups and the sulfoxy groups. And if you see them, you say, ha, this compound could undergo reduction of those functional groups. Reduction reactions can be abiotic or biologically mediated, but in almost every case, if a compound can go both abiotically and biologically, the biological reaction is going to be much faster, because living things like bacteria have little things called enzymes. And the whole point of an enzyme is to reduce the activation energy of a reaction, which makes it go faster. So the biological, if all the right conditions are available for bacteria to grow and do these kinds of things, then they will almost certainly carry out the reaction much faster than it could go via just an abiotic mechanism. But again, these types of reduction reactions require that you have some sort of anaerobic environment. And again, those could be things like deeply buried sediment or groundwater that has been flowing underground for a long time and has not had any opportunity to absorb any oxygen from the air, sewers, landfills, things like that where you have deeply anaerobic environments. And usually when this is happening, when it's happening biologically, what's happening is the bacteria are using that chlorinated or brominated compound as their electron acceptor. Because if we go back and look at the equations, my little buttons are just not working. Nope, not working. Not working at all. There we go. So if we want to reduce this by getting rid of some of the chlorides, we have to add electrons. So in other words, this compound is accepting these 2 electrons. And hopefully you've learned from your biological processes class that every living thing needs an electron donor and an electron acceptor. And it's electrons being taken away from the donor and given to the acceptor that's called the electron transport chain. And that's the thing that generates or releases the energy required for the living thing to grow. Okay, so the bacteria may be using these chlorinated or brominated compounds as electron acceptors to survive in part of their electron transport chain. I don't know why the little buttons are not working today. There we go. There are a small number of functional groups that can be abiotically oxidized. Those include things like mercaptans, anilines, and phenols. But generally, I'm not going to hold you two responsible for knowing that. Mercaptans, anilines, phenols. I'm not going to hold you responsible for knowing those, but it's something useful to log away in the back of your head in case you become a real environmental chemist and need to know those kinds of things. Okay, now in this class in 2020, because of COVID, we ran out of time and we did not have a chance to talk about aerobic biodegradation. So this is the only slide you're going to get on it. Some of you guys have spent more time than others studying degradation. You might have an advisor who is Donna Fennell or Lily Young or one of those people who does a lot of biodegradation work. So you know more about this than I do. But I will just say, a few words about it here. First is that most compounds can be aerobically degraded by bacteria. The rates might be slow, but they can be degraded. So, for example, PAHs are just polycyclic aromatic hydrocarbons, just a bunch of benzene rings stuck together. They can be aerobically degraded by bacteria. If you take some lab culture, some sludge from a wastewater treatment plant and put it in a serum bottle and add some PAHs, the PAHs will go away. That's what happens. But in the real world, the rate at which that happens is quite slow. So, for example, if we did a whole mass balance on PAHs in the New York, New Jersey harbor, we would assume that there was no biotransformation, and we'd probably be just fine because any transformation is pretty slow. The presence of chlorines and bromines and other halogens tends to block the aerobic degradation. Chlorines and bromines block this aerobic biodegradation, which is why PCBs, although they're just two benzene rings stuck together, if that's all they were, they would be pretty biodegradable. But when you add chlorines onto the rings, they basically block them. They act as a wall that just prevents the bacterium from getting in and attacking and eating up the juicy ring in the middle. So most enzymes for bacteria that attack aromatic rings attack at two adjacent positions on the ring. This is true for the kinds of enzymes that metabolize and eat the organic phenyl, aromatic rings, but it's also true for the cytochrome P450 system, which is the system in mammals and even in lower organisms like worms and fishes, that is supposed to detoxify a lot of these weird xenobiotic compounds that get into the organism. So the cytochrome P450 enzyme needs two adjacent positions on the aromatic ring that are not substituted, that don't have any chlorines or bromines or nitro groups or anything on them. And if it has those two open positions, typically the enzyme can get in there, attack, and break open the ring. But if all those positions are blocked, then biodegradation of aromatic rings will be slowed or stopped. And catabolism is a special word, a big fancy word. Catabolism is when the bacteria use the chemical for food and actually mineralize it. So they eat it all up. But something like those reductive dehalogenation reactions we were talking about is not catabolism. That's where the bacterium is using the halogenated compound as their electron acceptor, not an electron donor. Food is the electron donor. That's the jelly donuts and the sugars and the proteins and the fats that you eat. That is your food, and that is your electron donor. Your electron acceptor, because you're a human being, your electron acceptor is oxygen. Right, you guessed it. Okay. So higher animals like us, we transform chemicals in order to render it more water soluble and therefore hopefully less toxic. And so when I said that enzyme gets in there and breaks open the ring, it can also add hydroxyl groups to the ring, and that will make the compound more polar. If you add a hydroxyl group to an aromatic ring, you've created a phenol, which could become deprotonated and be negatively charged, and therefore it will be much more water soluble, and you'll be able to eliminate it in your urine. So the cytochrome P450 system is supposed to detoxify these nasty xenobiotic chemicals, but in some cases adding that hydroxyl group actually makes the chemical more toxic. That's what happens for PCBs, for example. If you have PCBs with only one or two chlorines on them, the cytochrome P450 system will go and add hydroxyl groups to it, and that actually makes the PCB molecules more toxic and not less. But that's just one example. Okay, now the other type of oxidation that we talked about was photolysis. Now, you will remember from the photolysis lecture that we didn't have to explicitly draw a lot of electrons changing hands. because we had a lot of free radical oxidation steps, lots of free radicals. Whenever you have free radicals, really you have oxidation going on. And if you sat down and really mapped out the oxidation states of everything that was happening in these reactions, you would see that oxidation states are changing. So direct photolysis can occur on only certain types of molecules, right? Only a few select classes of molecules will undergo direct photolysis. Direct photolysis involves absorbing the sun's energy by the chemical, which causes electrons to be promoted from the highest occupied molecular orbital to the lowest unoccupied molecular orbital. So the energy gap between those two molecular orbitals is the determining factor in how easily that electron can be promoted. And that energy gap ideally would be small. The smaller it is, the easier it will be for the electron to get promoted. So we said that the easiest way, the smallest energy gap occurs when you have lone pairs that are right next to pi bonds, like double bonds or aromatic rings. So the lone pairs are in n orbitals, and the pi bonds and aromatic rings are in pi orbitals, and you get an n. So if you have a pi, then you also have a pi star, right? Anytime you have a pi, you have a pi star. And so the transition goes from n to pi star, and that's a very relatively small energy gap. So chemicals that have lone pair electrons right next to double bonds or aromatic rings are liable to undergo direct photolysis. If you don't have that kind of structure, then you have to ask yourself, well, maybe my compound can undergo indirect photolysis. And most compounds can undergo indirect photolysis. If they have any kind of carbon-hydrogen bond, they can undergo direct photolysis. So really almost anything can undergo direct photolysis unless it's already completely halogenated, like Freon 12, which is dichlorodifluoromethane. It's already completely oxidized. It doesn't have any carbon-hydrogen bonds. It can't undergo indirect photolysis. But almost everything else in the world can. Indirect photolysis is going to be faster when it happens in the gas phase. In the water phase, the turbidity screens out a lot of the light, and things move more slowly in water because it's much more viscous than air, so that slows things down as well. So indirect photolysis and direct photolysis too are going to be more important in the gas phase, but they can happen in the aqueous phase as well. All right, so that is the end of our review of the different kinds of chemical reactions we talked about in the second half of the class. What I would encourage you to do for practice is to go look at all the chemicals that we may have talked about this semester. Look at the chemicals that were listed in your midterm exam, for example, and in the 2 quizzes that you took. Look at all those chemical structures and ask yourself, how would this chemical react? Go through this PowerPoint and look at every type of reaction and ask yourself, ask yourself, go right up here to the beginning and ask yourself, is it an acid or a base? Here's how you would figure that out. Does it undergo hydrolysis? Does it undergo SN2 or dehydrohalogenation? Does it have one of these structures, which can result in hydrolysis of carboxylic and carbonic acid derivatives? Does it have halogens that can undergo hydrogenolysis? Does it have halogens on 2 adjacent carbons that can undergo reductive elimination? Does it have nitro or sulfoxy groups? Is it one of these guys that can be oxidized? And can it undergo either direct or indirect photolysis? And practice that with as many chemicals as you can think of. And that is probably the best way to prepare you for the final exam. So this is it, guys. Have a great summer. And whenever you have questions about environmental organic chemistry, you can always feel free to email me or go back to your textbook. I hope that what you've learned in this class has been really helpful and will continue to serve you well for a long, long time.